Okay, I think we're ready to start. So, hello, a third time for those of you who logged in early. Thank you for that. I'm Elad, the CEO and co-founder of Astrolabe Diagnostics, and I am very excited to host Adeeb Rahman from Mount Sinai today. Before we begin, we're actually going to do a bit of a sound check, so to speak. If you have any questions during the webinar, please use the Q&A button below. Just click on it and type in your question. And just to make sure it's working, how about you go there now and type your favorite marker, I guess. Let's see how many CD3s we get. So just go to Q&A and type in your favorite flow cytometry or mass cytometry marker for a bit of a survey. And I'm going to have to stop sharing to actually see everyone's results. So I'm seeing CD45, CD69, CD169, CCR6, another CD45. Yeah, I should have said CD45. CD45 is a much more sensible choice. Yeah, I think CD45 is the winner here. Already 4 votes. CD200, AGL-ADR. Great. So it looks like the Q&A is working. Thank you all for participating. Again, if you have any questions, just use the Q&A button below. And I'm now going to pass the mic to Adeeb. Adeeb Rahman is an associate professor of genetics and genomics at the Icahn School of Medicine at Mount Sinai. He also serves as the director of technology development for Sinai's Human Immune Monitoring Center. I'm sure many of you are familiar with Adeeb in that role, where he oversees one of the world's most active full-service mass cytometry programs. And at this point, they processed over 10,000 samples over a wide range of basic translational and clinical research studies. And without further ado, I'm going to pass the mic to Adeeb. Thank you for joining us today.
Thanks a lot. Okay, so hello, everybody. Thanks for joining. So I'm going to talk today about a bunch of topics that are pretty in the weeds on the technical side of mass cytometry, because I think, as Elad mentioned, we've run a lot of samples here at the HIMC, and in the course of running all those samples, we've seen a whole host of interesting kind of issues come up. And I want to talk to you about a particularly interesting and I think sort of unusual one that's come up in some more recent studies. Before getting into sort of the details of that, I'm going to start with a little bit of sort of cytometry and science philosophy. Which is that obviously high-dimensional cytometry, be it flow cytometry or mass cytometry, is an extremely powerful tool to be able to analyze complex samples and discover sort of cellular phenotypes, cellular biomarkers. So an ideal sort of cytometry study is you're going to have a large number of interesting samples from interesting cohort. You're going to measure 40 proteins on millions of cells across all those different samples, run some sort of fancy analysis, and discover some complex, ideally never before described novel cell type that changes in relation to whatever perturbation you're looking at, or infection time course or what have you, and then you publish it, and then hopefully you also make your data public, because everyone should do that. So that's the trajectory that we like things to go down, but I think there's a strong focus, generally speaking, on that last part of this, where you have this interesting phenotype you've discovered. But in order for that discovery to be robust and reproducible, in designing the experiment to generate that data, you really need to be aware of and minimize potential technical confounders that could get in the way of that final discovery. So using this sort of hypothetical scenario here, which we could say is sort of an ideal or maybe like a ground truth of some sort of biological situation, there's a bunch of sort of alternative scenarios that can happen and are often more likely. So one is, you might have a true phenotype, but because of issues in how you've designed your panel or the assay you're using, let's say you're measuring the expression of a particular protein, but you have a not very sensitive reporter, you might end up sort of minimizing your effect size. So instead of being able to detect a relatively large delta, you have now a smaller delta. And so this can increase the risk of type 2 statistical errors, so essentially false negatives, so essentially concluding that there's nothing there when there in fact is something interesting there. Another scenario is, let's say you just have a lot of noise in your measurements due to technical variability, be it issues with sample processing or staining or things of that sort. So once again, you can end up with sort of type 2 statistical errors where, because of all this noise in your data, you're less able to to conclude what's changing robustly. And another and sort of equally potentially more risky issue is the opposing issue, where you can have some sort of confounding batch effect or off-target reagent detection that's going on in addition to the biological process. So let's say you still have this true scenario, but you ran all of your pre-samples in March, and then you ran all of your post-samples in November after you switched out the detector on your instrument, and now it's more sensitive. And so you now have a confounding issue where you may think that you're seeing something that's biologically driven, but in fact it's actually driven by some technical issue. And so this results in the opposite problem of type 1 statistical errors or false positives. So it's important to always keep these things in mind and to try and think of ways, where sources in your workflow might contribute to these kinds of problems and how you might think about mitigating them. And so every step in a cytometry workflow has some potential of contributing to potential issues. So at the time of initial sample collection, you can have issues with blood collection tubes you're using, or storage time, or initial sample quality. If you're doing tissue work, there might be the digestion enzyme you're using. If you're cryopreserving samples for later analysis, the cryopreservation protocol, your post-flow viability, these are all things that can contribute technical artifacts that can get in the way of your true biology. At the time of antibody staining, another major source of error is basic things like pipetting errors. We've definitely had large-scale studies where we discover that some marker looks like it's changing, and we can track it back to just issues with a pipetting problem, or protocol variations over time, particularly in longitudinal studies. Reagents degrading over time, lot-to-lot variability in those reagents. During the actual data acquisition, there's the instrument performance, inter-instrument variability. If you're trying to do, let's say, a multi-site study, or if you even at a single site, we have 3 Helioses, for example, and they all have particular quirks and behave in particular ways. And so comparing data between instruments can be a challenge sometimes. And then during the course of acquisition, even on a single instrument, you can have weird sample degradation artifacts or things that happen. So like this example plot here, you can see there's sort of signal drift happening over the course of this acquisition where the negative is rising. So again, another potential source. And then lastly, also with data analysis. So if you're drawing gates and you have different people doing those analyses, or even the same person, and they're tailoring gates for different samples, where that gate gets drawn can have some impact on the downstream results. So there's obviously many sources that can introduce unwanted variability into potential experiments. And there's also similar ways of addressing all of these sources. So I think one of the first things is just to be very aware of them and to make sure that you're thinking about them and you're designing your experimental protocol to be able to mitigate them. So kind of working backwards here, for data analysis, you can think about having standardized and transparent data processing analysis pipelines on the instrument side, making sure you optimally maintain and track everything. And then for kind of the first two processes, the key part there is you want to try and standardize and simplify your sample collection and processing workflows as much as possible. Like generally speaking, the simpler a workflow, the fewer steps there are for some sort of unintended variability to be introduced. And similarly with antibody panels, standardizing those antibody panels as much as possible can also really help to mitigate some of those issues. And so in regards to those first two steps, one of the things that we've really been a big fans of at the HIMC is using lyophilized antibody panels. And we've been doing this for a couple of years now, especially in many of our large longitudinal studies. And we've been building our in-house lyophilized panels to do this kind of work, which is sort of a lot of work in its own right. It has a high burden of sort of validation to be able to generate these panels, and a big kind of financial upfront cost in terms of generating them as well. So we were actually very excited when Fluidigm released their own commercial lyophilized panel. I believe it was last year, maybe the year before, but the Fluidigm MaxPar direct immune profiling assay. So this is kind of a complete kit. It includes kind of a one tube, like a dry tube of antibodies that contains 30 antibodies that span all of the major kind of phenotyping markers that are relevant for looking at circulating immune cell populations. Allows you to identify kind of all these populations highlighted on the right. And it also has room for additional drop-in antibodies. So kind of a very nice solution there that's kind of provides a one tube format for doing cell profiling. And it's amenable for both PBMCs as well as whole blood. And we did a lot of work focused on sort of applying it for whole blood, and we made some kind of additional modifications to the protocol to facilitate its use in multicenter studies and also in relation to some of the COVID work that we've been doing. So we started a very large institutional biobanking effort of samples from hospitalized COVID patients during sort of the first wave of infections in March in New York last year. And this is really kind of an issue of I'd say doing high throughput cytometry under siege, so to speak, right? Because we were kind of in the midst of a shutdown. The cases were out of control. We had a lot of patients in the hospital, and we recruited this army of volunteers who were from all across the institution. People essentially who had been furloughed from their positions, but were still wanted to be contributing to the effort. And so lots of folks who were helping to collect samples from these patients and bank them and process them. And we wanted to include a mass cytometry profiling as part of this effort. But this essentially was a large group of non-specialists, and we didn't want to have complex workflows and have to train people when they also had a lot of other things to do. Plus, everything was happening under BSL-2+ conditions, and so for those of you who've worked in those conditions, you're probably aware that they're fairly uncomfortable, and you kind of would prefer to not be in those conditions as much as possible. So we wanted a really streamlined workflow that minimized the amount of upstream processing that needed to be done. And so, in contrast to sort of a conventional CyTOF workflow highlighted here in one, the MaxPar Direct already provides one major advantage in that instead of having to worry about managing a large batch of reagents and dealing with 30 antibodies, you can now condense this down to a single addition to one antibody tube. So that helps a lot. But we wanted to simplify this even further, and we did so using this smart tube-based blood stabilization protocol. So nothing super profound. But the idea being essentially that in protocol 4, we have a workflow whereby you can take blood, add it directly to a tube, incubate the blood, add the stabilizer, and then just stick your tube at -80. And this has two advantages. One, it allows for just 2 pipetting steps up front at the very beginning. Then the stabilizer also ends up actually neutralizing, because it's a formaldehyde-based and a fairly high concentration formaldehyde-based stabilizer. So it sort of neutralizes any residual virus, and that allows the tube to just be put away. And then after the storage period, the HI&C team can now come in and process large batches of samples and add in additional antibodies and do all the extra downstream work. So this was really a kind of a very helpful workflow in allowing us to be able to run a very large number of samples kind of very quickly without sort of having kind of a big team in place to be able to do that upfront processing. I will note there's an even sort of faster workflow whereby you could just take your blood and stabilize it up front and then stain it with the MaxPar panel later. The problem with this is that there are several antibodies in the panel that are fixation sensitive, and you'll lose expression of those down the line. And this is all published work now. Actually, the paper just came out in Cytometry A 2 days ago, so you can check it out there. It's also in medRxiv, showing all of the sort of optimization that we had done to be able to kind of confirm that this workflow is a viable workflow. And we've also applied this then to study COVID patients. So, for example, just showing you some example data here from some healthy donors, some moderate patients, hospitalized patients with moderate disease and those with severe disease. And this kind of shows one of the nice advantages of whole blood versus PBMCs, in that you get to analyze the granulocyte compartments, so your neutrophils and eosinophils. And certainly in the case of COVID, elevated neutrophil counts and changes in neutrophil phenotype are a very sort of defining attribute both of infection as well as the severity of infection, with concurrent reductions in T cell populations. And there's several other more defined cell types that can be identified with the panel that we were able to show differences for. And we're now in the process of, we've taken the same workflow and we're applying it as part of an even larger study, the NIH-funded IMPACT study, which is a multi-site study across, I think, 12 or 13 clinical sites across the country, where again, we have a bunch of sort of non-expert individuals collecting samples. And this has proven to be a very kind of convenient workflow for allowing them to incorporate mass cytometry into the study, even though most of the sites don't have their own instrument, because essentially they can collect the samples, bank the blood, and then ship it to us as a central site to do all the final processing. So all in all, a fairly powerful workflow. So that's the good news. But now getting to sort of the weirdness that I wanted to focus on for some of the rest of the seminar, which is that in the context of running these very large numbers of samples stained with this panel, we started noticing kind of a weird staining artifact in some samples. So here's one example of what a normal staining expression should look like. So just 2 markers that we're all probably very used to looking at, C19 versus CD3. CD3 is your T cell marker, CD19 is on your B cells, and you can see that the B cells highlighted in orange up here at the top left are CD19 positive. Your T cells are CD19 negative. And then there's obviously a lot of multiplets that happen in mass cytometry, and so the few kind of dots you see in the upper left quadrant there, upper right quadrant there represent multiplets. But that's all in all a pretty normal looking sample. But there were a subset of samples where we started seeing things that look like this, where we now have kind of this aberrant staining of CD19 that was appearing on non-B cell populations. So you can see a subset of the CD3 positive cells here that are expressing reasonably high levels of CD19. And we know that these aren't, in addition to just CD19, another antibody in the panel that we noticed some issues with was CCR6. So often in samples where we saw this kind of aberrant CCR or this aberrant CD19 staining, we would also be seeing co-expression of CCR6 with that same kind of strangely staining population. And both CD19 and CCR6 are expressed on B cells. So one question is just, is this some sort of weird B cell population? And it appears not to be, because if you look at something like CD20 in the panel, that staining is quite clean. So the cells essentially that are staining appear to be non-B cells in that they're CD20 negative, and positive for a bunch of other markers. So another way to kind of dig a little deeper into the phenotype of these cells is just doing some dimensionality reduction. So here is a t-SNE analysis of a stained sample. So I'm excluding the neutrophils and eosinophils here just to focus on the kind of PBMC fraction of the sample. And I've excluded CD19 as a clustering parameter for this analysis. But if we now look at these populations we've defined, and now we overlay CD19 expression, you can see donor B here, who's an unaffected donor. As expected, the CD19 is really localized just to the B cell compartment, whereas in an affected donor, you can see that once again, we are seeing CD19 on the B cells, but we're also seeing this sort of CD19 distributed across a bunch of different cell types, including sort of NK cells, gamma delta T cells, subsets of other T cells, and specifically kind of the naive T cell populations. Here you can see kind of in the CD45 RA high naive CD4s, for example. And so to quantify the prevalence of this, because we know that essentially CD19 staining on naive CD4 T cells is kind of one of the attributes of this artifact, we can kind of quantify that across a much larger number of samples. So this is kind of MDIPA stained samples, probably, I forget what the N is here, but I think it's probably in the 200 plus sample range. And if we look at the expression of CD19 on the CD4 T cells, about 20% of the subjects have sort of what I would define as kind of elevated, unusually elevated levels of CD19, the extent kind of varying. So only a minority of patients have extremely high levels, but several samples where we see, higher staining than would be expected. So yeah, kind of unusual. And so what's going on over here? So one thing that we determined was, this appears to be a donor-dependent issue. So as we said here, it's only about 20% of subjects tested. But interestingly, among those subjects tested, they were fairly consistent. So this is an example of one affected subject where we've collected replica blood samples from the same individual several weeks apart, stained with independent lots of the MDIPA, and we can consistently see this phenotype. And there's some sort of lot-to-lot variability with the different lots of the MDIPA kit, but generally speaking, the phenomenon is kind of present in an individual who is affected. But interestingly, we had PBMCs banked from this subject at T1. They've been collected in parallel. And when we went back and stained those PBMC samples from this subject, they were completely normal. So there's no evidence of the artifact there. And so again, whole blood seems to be affected, PBMCs not affected. So this really suggested that it was something to do sort of a whole blood specific artifact. And so in thinking about what are the differences between whole blood and PBMCs, one aspect is just the presence of these granulocyte populations, which get removed by Phi calling, but also the fact that whole blood staining is generally done in the presence of endogenous plasma, whereas PBMCs have been washed and banked, and now we're usually doing the staining just in, FACS buffer, FBS, something of that sort. So to investigate the possibility that plasma factors were leading to this artifact, decided to do this sort of interesting sort of sample swap experiment where we got blood from an affected donor and an unaffected donor, spun it down and collected the plasma fraction, and then took the remaining blood cells and extensively washed them to remove any endogenous plasma, and then kind of did this matrix swap where we were staining blood from an affected individual in the presence of either autologous plasma or heterologous plasma from an unaffected individual, and vice versa. And in doing that experiment, this is what the data looks like, and it's pretty clear, where you can see the top row here is showing blood cells from the affected individual. The bottom row is showing blood cells from an unaffected individual, and then the different columns here are showing different staining matrices. So you can see if you take those blood cells and you stain them just in the presence of PBS, the artifact is completely gone. The CD19 staining here is now localized just to the B cell compartment, as you would expect. If we now stain them in the presence of plasma from the affected donor, the artifact remanifests and it's transferable. So now the unaffected donor cells are showing this affected problem. And then reciprocally, if we stain the blood in the presence of plasma from the unaffected donor, the affected donor, again, no longer has the problem. So this isn't a cell intrinsic artifact. It's nothing to do with the cells themselves. It really does seem to be stemming from some sort of factor in the plasma of these sort of affected individuals. Pretty unusual. And so then the question was, this is pretty weird. Like, how is a plasma factor affecting CD19? The CD19 epitope is pretty well established. It's pretty well studied. It's kind of, things like this haven't really been reported before. And kind of the short answer is it's sort of not really a CD19 issue. So a few pieces of evidence for this. So despite the fact that the aberrant staining is being seen in CD19, that seems to be more of a symptom than a cause. So one issue here is that this seems to be an MDIPA specific artifact because, as I mentioned at the start, we also have in-house lyophilized panels that we've generated, and we've never seen any evidence of this using any of our in-house panels. So here's an example here, again, showing that those samples stained with the MDIPA panel on the left, where we see some evidence of the staining, and our in-house panel on the right, where we really don't see the staining. So already suggesting that it's not necessarily a CD19 issue, but to kind of definitively address this even more carefully, we did this experiment where we took some whole blood from a known affected donor, and in addition to staining with the MDIPA panel, we actually spiked in a CD19 PE antibody, and this is the same CD19 clone that is in the MDIPA panel. So essentially the identical antibody conjugated to PE, and then we could come in with an anti-PE antibody, and look at this. And when we do this, you can see that the CD19 staining is localized. So the PE staining is localized specifically to the B cell compartment as expected. So as we would expect, this particular clone is now only interacting with the B cells. And in contrast, if we look at the CD19 in the context of the MDIPA panel, once again, we see the staining on the B cells, but we see this aberrant staining. And when we introduce this competing CD19 antibody, we see that there's a reduction in intensity on the true B cells. So essentially, this represents bona fide competition for the limiting epitope between the two antibodies, whereas there's zero effect on the aberrant staining pattern. And this is just another way to show that data. Gating on these different populations, you can see that if you look at the staining of the MDIPA CD19 on the B cells, it goes down a little bit in the presence of the competing antibody, but none of the aberrant staining is affected. So this really definitively shows that this is unrelated to the antibody clone or the epitope or the paratope. So again, the epitope being the antigen that's being recognized, the paratope being the portion of the antibody that's recognizing that epitope. And so none of those are affected. So this isn't a CD19 off-target staining. But interestingly enough, and this almost like stumbling on this, in doing these experiments, I just happened to notice this sort of phenomenon whereby the aberrant CD19 staining, so essentially the pattern of CD19 staining outside of the B cell compartment, is sort of phenocopying the distribution of CD45RA. So here you can see CD45RA staining, and here you can see the CD19 staining. And so if you kind of ignore the B cell compartment where we know there's CD19, you can sort of see that this kind of off-target CD19 is almost like a shadow of the CD45RA. Like you can see here, these populations that are high for 45RA are also high for CD19 and vice versa. So to try and sort of address this, we did that same sort of experiment with the blocking antibody, but now we added a blocking antibody against CD45RA. And so when you stain the sample with CD45RA APC together with an anti-APC, the resulting CD45RA APC pattern matches the expected CD45RA pattern that you see in the MDIPA panel. But by doing this, we're also now kind of out-competing the MDIPA CD45 staining. And interestingly, by blocking the CD45RA staining, we also block the aberrant CD19 staining. So you can see that even though we haven't done anything to the actual CD19 stain in the panel, simply adding in that CD45RA has now also removed this weird data artifact. And just another way to look at this data, for those of you who are more accustomed to regular biaxial plots. So here is looking at CD45RA in the MDIPA versus CD19. You can see this kind of diagonal here of the kind of correlated expression of the two markers in the non-B cell compartment. And now when we come in with the CD45RA APC, we no longer see that relationship, and that off-target staining is kind of removed. And I think I mentioned right at the start that the kind of weird CD19 staining that we were seeing appeared to be associated with also some weird CCR6 staining. And interestingly, yeah, blocking the CD45RA interaction also seems to be minimizing that CCR6 kind of off-target staining, which you can kind of see over here, and is now gone over here. So, all right, so what's going on here? Kind of putting all this together. And yeah, my disclaimer here, there's a lot of major hand waving to follow. This is just kind of me spitballing with what I think might be going on. So within this lyophilized panel, there's 30 antibodies. And I suspect that Fluidigm is using a range of different polymers in generating this. And I know this for sure because Fluidigm doesn't disclose what polymers they use on what antibodies in their pre-labeled antibodies. But one of the pieces of evidence that suggests this to me is that if you look at the quality of CD19 staining in the context of the MDIPA panel, the median CD19 intensity on B cells is much, much higher than anything we've ever been able to achieve with any of our in-house conjugate antibodies using either the X8 or the DN3 polymers, which are the two kind of commercial polymers that Fluidigm sells for user conjugations. So, and this is something that's not solvable by titration, right? CD19 is a relatively low-ish density antigen, and so throwing more antibody at it ultimately has diminishing returns. And the only way that you can really boost that intensity is to use some sort of a polymer that's able to add more metal to a given antibody. So I suspect that's what's happening in the panel. So I think that there's some antibodies in the panel that are using kind of polymers that are different from other antibodies in the panel. And now, in some donors, there is now some donor-specific plasma or serum factor that's somehow kind of cross-linking or interacting with this polymer that's present in only a subset of these antibodies. So in this case, 45RA19, for example. So what this serum factor might be remains a mystery. And I've kind of spent a while poking into this, and I'm still not sure. I mean, I think the most kind of crazy thing we've tried doing is in a subset of our COVID cohort where we saw this artifact, we were also fortunate enough to have done SomaLogics as a parallel assay. So for those of you who aren't familiar with that assay, it's essentially an aptamer-based proteomic profiling assay where we're profiling the expression of over 2,000 different proteins in the plasma of these patients. And I tried running just like a quick correlation between every single one of those 2,000 plus plasma factors and the presence or absence of this artifact, and couldn't find any factor that meaningfully related. So that's kind of as much effort as I put into this, and I'm still not sure what might be going on there. So mystery factor. But now, as a result of the presence of this mystery factor, when you take this panel and you try and stain cells, if you now have a cell that is CD45RA positive, when that CD45RA antibody binds to the cell, it's essentially bringing on the 144ND CD19 antibody along for the ride, ending up with sort of this dual staining of two metals at the same time. Another sort of intriguing possibility is that maybe this actually doesn't relate to the actual antibody epitope or paratope, but it could also be a direct interaction between 45RA and some aspect of this complex. Because so as far as I know, CD45RA doesn't actually have a known ligand, but it's a highly glycosylated protein, suggesting that it could potentially interact with lectins, for example. And so if this donor-specific factor is some sort of lectin that's binding things together, maybe there's a direct kind of lectin interaction going on with the 45RA molecule. So, and then one question is, why is 45RA the issue here? Because technically the reciprocal could also be happening. If this hypothesis is true, you would expect that an interaction with CD19 would also bring 45RA along for the ride. The problem there that's harder to tell is that B cells already are 45RA positive, so it's kind of looking at positive staining on something that's already positive. And then the bigger issue was also one of antigen density. So it turns out that CD45RA is one of the highest density antigens that we typically measure in flow cytometry. You can see here on this log scale, it's kind of an order of magnitude higher than many other antibodies. I actually don't know if CD19 is on here. Oh, it is, yeah, down here. So you can see it's several orders of magnitude higher than CD19. So if you assume that this kind of cross-linking is only happening with kind of a proportion of the antibody, even a small proportion of a fraction of the 45RA staining could end up giving you a detectable signal, whereas sort of a fraction of a weaker stain is more likely to be kind of below the limit of detection of the CyTOF or low enough along the limit of detection to not really be manifesting in sort of a meaningful way in the data. So that's what I think might be going on there. So regardless of how exactly this is happening, how do we solve it is the bigger question. And I think the experiments that I have shown you so far already, I think, provide some useful insights into how to potentially tackle this. So one option is obviously work with PBMCs, because we don't seem to see the problem there. And if you do want to work with whole blood, because you want to look at neutrophils or eosinophils and or other reasons that you might want to work in whole blood, using washed whole blood seems to be an easy way to mitigate this problem. So removing the presence of the endogenous plasma essentially removes this donor-specific serum factor, and now this is no longer an issue. The downside of that, obviously, is that it does complicate the upfront workflow a little bit. One of our goals here was to really minimize any sort of upfront steps before you start the process. And so either isolating PBMCs or kind of washing the whole blood sort of defeats the purpose there a little bit. So another option that you could do if you want to just use straight whole blood is you can potentially just spike that whole blood with a competing CD45RA antibody during the MDIPA staining. And you could either do this coupled to a different channel, or you could even use CD45RA coupled to 150 just with a different polymer. So essentially you displace the MDIPA 45RA and you essentially substitute it with your own CD45RA in the same channel, which should hopefully kind of prevent the interaction of that entire complex. And then essentially the fourth option is that Fluidigm fixes the problem, which would really be the optimal solution, hopefully. And they're aware of this. We've been in close contact with them for several months about the issue, so they know it exists. They reproduce it on their hands, and they are, I think, trying to address it because more recent lots of the MDIPA kit that we purchased suffer less from this issue, so they're screening samples on their end. But it's still something I think to be aware of and watch out for in case you run into it. And then a third question is, what do you do if you've already run samples, you weren't aware of this, and you now have data where you're seeing this issue? What should you do there? So I think, the first and most important part is hopefully what I've convinced you now with the data I've shown you is that this kind of, the CD19 staining is, it's a technical artifact. So it's not like the cells that appear to be CD19 positive, you don't need to necessarily discard them from your analysis. Your patient probably doesn't have lymphoma, unless they do have lymphoma, in which case this is probably a confounder in determining that. But you need to think about, if you're doing manual gating, for example, and you're gating your T cells, you don't want to use CD19 as an exclusion parameter when gating your T cells. And so it really becomes more of an issue if you're trying to use automated analyses or clustering or things of that sort, where you don't have as much control over some of those factors. And so just to show you one example of this, just using Astrolabe. So here's an example of a sample where we really don't see the issue, CD19 with CD3 there on the top. And if we just do some basic manual gating of major populations and we compare this to sort of major populations identified using the Astrolabe, like standard hierarchy, we see extremely good correlations, as we've seen in many other contexts. But if we now take a sample where we do see this artifact, and this is admittedly like one of the worst cases of this that we've seen. This is actually not our data, it's data that was sent to me by a collaborator who had also encountered the same problem. And now if we try and run sort of Astrolabe versus manual gating, the correlation is much worse because Astrolabe doesn't really know how to deal with this population that's kind of co-expressing CD19 and a bunch of other markers. But a very simple fix here is we just reran the same sample using a custom hierarchy where CD19 is now dropped from the standard definition of any cell type, and this essentially restores our correlation back to being pretty good again now. Because now that these cells that appear to have this aberrant CD19 staining are essentially being defined based on the other markers in the panel and not CD19. So fairly addressable. I guess one thing to note is that the CD19 issue is quite addressable computationally, largely because CD19 is such a well-established marker. It has such restricted expression. Like, we really know what it's supposed to look like, outside of maybe the lymphoma context. The CCR6 issue I mentioned is actually a little bit harder to tackle. One, because I think it's actually a little more prevalent, a little more subtle. And two, it is promiscuous. It's expressed in a bunch of different cell types. So in general, I think it's safest to just exclude this marker from analysis if you find a sample that is affected by this. I haven't found a great computational method of determining kind of true CCR6 expression versus potential artifactual CCR6 expression in those samples. All right, so to kind of conclude and wrap this up, so cytometry, be it flow cytometry, mass cytometry, CytSeq, all of these assays are based on indirect protein measurement, right? So we care about measuring the expression of CD3 on a cell. If you have a cell that has a protein, you're using an antibody with a detector on it, some sort of detection modality for that reporter, and then you end up with data. And so when we do this, everyone sort of focuses on the data at the very end, right? And we say that this t-SNE plot here, or UMAP in this case, is showing expression of my protein, X, whatever it is. But there's a lot of kind of implicit assumptions that are going into that. So you are really hoping that your protein is directly correlated with your antibody. So your antibody is a surrogate for the protein, the reporter is a surrogate for the antibody, your detector is accurately picking up that reporter, and thus your measured value is hopefully a direct measure of that protein, right? But these are assumptions, and technical artifacts can undermine those assumptions. And so to do good science, you need to always be aware of what those assumptions are. Don't take them for granted. You need to consider what are factors that may impact the validity of those assumptions, and then also how this may ultimately impact your interpretation of the data. And so it's always good to keep this in mind, just to do good science. And just to show you, some examples of things that can happen. So every step of this path has certain technical issues that can impact those assumptions. So let's say you're doing regular flow cytometry. Cellular fluorescence or fluorescence spillover, or cellular autofluorescence, fluorescence spillover, is an example of your detector measuring a signal that's not actually attributable to the reporter you think it is. So if your cell has autofluorescence and you're seeing signal in the FITC channel, it's not actually FITC that's causing that signal, right? It's autofluorescence. Same problem with spillover, and likewise in the mass cytometry world with isotopic impurity or oxidation, right? So if you're measuring a signal in 160 gadolinium, which is conjugated to some antibody, but there's some spillover coming from the -16 channel, you may inadvertently be attributing this signal to the 160 gadolinium reporter when it's actually coming from something else. And similarly, instrument issues. So if you have variations in your sensitivity over time, you might think that you're seeing differences, and it's actually just the instrument varying its performance. So these are all scenarios where it's not excluding it entirely. So it's not that the correlation doesn't exist, but you have other factors that can be impacting your assumption that what you're measuring actually reflects your protein. So going down the line, there's situations where your antibody can sort of become uncoupled from your reporter. So let's say you have a poor conjugation, and now you have a lot of free metal floating around in your prep. So that free metal is now going to be detected in the absence of the antibody, and is also going to lead to sort of a break in this correlation. Let's say your reagent is degrading. Let's say you're using a tandem dye, and the tandem is now splitting, so your APC is uncoupled from your size 7. Things like that can happen. Or let's say you have metal contamination in your reagent, in your sample for some reason. The patient happened to have received cisplatinum therapy, and now you're measuring platinum, and so you have a metal that's being measured independent of the affinity reagent that you're using. And then also, of course, there's this issue of off-target binding, which is kind of what today's talk is sort of focused. And again, there's various flavors of off-target binding that are kind of well established. We all know, hopefully, about FC-mediated antibody binding, which you can mediate using FC block, standard part of most cytometry workflows. In addition, like a whole blood specific problem, especially when you're doing intercellular staining in mass cytometry, is charge-based interactions. So we previously published this showing that eosinophils, for example, will non-specifically bind to CyTOF antibodies due to their highly cationic granules. And we found that using heparin as a competing anionic molecule can mitigate that issue. And so today, I think the issue I talked about is kind of what I believe is a completely kind of new class of off-target antibody binding. I don't think has really been reported before. And it seems to be, again, specific plasma factors in specific donors and specific antibodies in this MDIPA panel. But the implication might be this may not be an MDIPA specific issue. It could potentially be kind of a polymer related issue, depending on what those polymers are and whether they're being used in the context of other Fluidigm antibodies outside of the context of the MDIPA. And yeah, so I think hopefully everyone can sort of keep an eye out for this and see if this is something that does appear in other contexts. All right, so I'm going to end there by acknowledging the whole HIMC CyTOF team, in particular Daniel and Brian, who did a lot of this work. Thank all of our blood donors, in particular, the individual donor A here, where after we discovered the person was a susceptible individual, they're probably wondering why we were haunting them like vampires and asking for repeated blood donations on a regular basis to kind of dig into this. So thank you all of them. The COVID work I showed at the very start was part of a very large institutional effort with a lot of volunteers and the entire Mount Sinai COVID biobanking in-house team. Also the Fluidigm technical team, who were very helpful in bouncing ideas off of when we first encountered this, and they did provide us with a free MDIPA kit to do some of this troubleshooting. And then all the work I present today is actually out in two preprint in the paper for all the COVID work, and a preprint specifically digging into this sort of donor-specific artifact that I mentioned. And right, I'll stop there, and hopefully that'll leave some time for questions.
Thank you, Adeeb, for this in-depth work. And I think that we at the CyTOF community are very lucky to have you an ambassador for the technology. And just a reminder, if you have any questions, please use the Q&A button at the bottom of the screen. Just type it in, and Adeeb will answer it. While we wait for people to ask questions, I actually have a question. I think one of the trends we've been seeing since the CyTOF was first released is that there's a productionization of many of the elements of the assay. So there's off-the-shelf solutions for different kits, and for the analysis, there's dimensionality reduction and clustering. I think it's typical for people to grab an off-the-shelf kit, run it through an off-the-shelf bioinformatics pipeline, and look at the results. Do you have any suggestions for QC steps that can be used in future experiments to catch similar issues to what you described today?
Yeah, that's a great question. And it doesn't have an easy answer. But I think one of the, and this is again just part of my general scientific philosophy, is it's important to be skeptical of data, right? Like your assumption, rather than being sort of credulous and sort of believing everything you see the first time, is to always think about, is what you're seeing really being driven by what you think you're seeing? And I think it's not a direct consequence, but I think often when things are hard, right, when commercial solutions aren't easily available, technology is generally accessible to fewer people, and the people who generally are involved in technology at that early stage tend to have this mindset to a much greater degree, like myself included. And I think the real strength of, I forget the term you use, but like industrialization, I guess, maybe, but I think the really great thing about that is it really does democratize access, right? It makes complex technology much more accessible to people who might not otherwise be able to or be willing to engage with it at that level. So I think that's a great thing. But the risk is exactly as you've mentioned, you kind of have an inclination to kind of take things at face value and just hope they work. And so my general advice would be that to try and maintain the skeptical sort of mindset, regardless of how easy it was to get that general data. And I think particularly when you're using like fully automated analysis solutions. So I mean, like nice thing with even things like Astrolabe or Pathsetter is they really provide this ability to push a button and get an insight. And now you have sort of a frequency of a cell type that's been defined for you. But I think even in those situations, it's very important to make sure you sort of remain engaged with the raw data. And this is a feature I really like about Astrolabe. And one of the reasons I really don't use Pathsetter is the idea of encoding single cell definitions into an FCS file. So at a single cell level, telling me what you annotated every cell as, that's like a critical aspect for me, at least, in reviewing data analysis, because I want to be able to double check what an automated solution is doing. And similarly, when data is made public, and this also puts a greater burden on, I think, making data public, because an important part of making data public is maybe you don't have the expertise to catch all these artifacts, but at least if your data is out there for the broader community to evaluate, someone else might be able to find it and dig into it. And I think in being able to encode your annotations in the data that way is very helpful. So I think that's kind kind of a roundabout answer. Obviously, there are standard QC steps you can take, but you can only QC for things that you kind of know about. In finding things that are totally new artifacts, sometimes it's harder to know how to build a standard analytical solution.
Yeah, and I think that skepticism and transparency are at least some of the reasons I became a scientist. So hopefully others follow that route as well. I have another question. Great presentation, Adeeb, as always. Question regarding the strategy of dealing with the aberrant staining on the analysis end by dropping CD19 from the hierarchy. Do you see any issues with phenotyping B-cell subpopulations, specifically plasmablasts and plasma cells, since their CD20 expression may be variable to absent?
Yep, that's a great question, and it's absolutely a potential issue. The one thing I will say is that the plasma cells are very phenotypically distinct, and they're relatively easy to define using other markers. Particularly if you supplement a few other markers into the panel. So at least in most of our usage of the MDIPA panel, we happen to be also adding in sort of CD39 and CD71, and there's several other markers that end up defining the plasmablast population pretty cleanly. So, and I guess it's a double thing to be aware. So I think it is a problem again with automated solutions sometimes because certainly in the absence of CD20, a plasma cell can look a lot like an activated T cell with low CD3 expression, for example. So it is something to be aware of, but it's certainly a tractable solution.
And if I understand you correctly, in your case, it was easier since you had these additional markers for these populations. Okay, but these markers don't
made it easier by having additional markers. But even with the regular MDIPA panel, sort of CD38 and CD27, kind of in the absence of CD3, does an okay job of finding them.
Okay, that was my next question. So there is something in the MDIPA that helps with that.
Yeah.
Hi, Adi. Oh, sorry, go ahead.
I was going to say the one other thing also is that, I guess there's dropping CD19 entirely in a hierarchy, and there's also, maybe thinking about how you might want to include it potentially, because the idea is that the true CD19 staining is still there. There's just other CD19 staining. So if you do get your plasmablast, you would hope that they should still be CD19 positive. So it's kind of an additional confirmation you can do that they are what you think they are.
Yeah, and I imagine that another strategy is to first label on the myeloid and CD3 and CD20 markers, and then out of the lineage negative, you can look at CD19 and
Yeah.
Hi, Adeeb. Thanks for this great talk. I have an off-topic question. Did you measure these samples with the WB injector or with the HT injector?
This was all wide-bore injector. Yeah, so essentially on our instruments, you guys may have heard prior seminars I've given on the whole wide-bore, narrow-bore thing. And we have a paper out on that as well. But at least for our helioses, they are essentially not usable with a narrow-bore injector, in my opinion. Like the data quality is just too bad. So we all of our instruments use the wide-bore injector. And that does seem to be an instrument-specific issue. So not everybody's instrument is in that category, but at least for us. Yeah.
And if I recall correctly, we had a whole webinar about this question. So whomever asked, you're welcome to look at the YouTube channel for Astrolabe, and I think we have a video there about that. Do you know if these donor-specific abnormalities are reproduced in spiked-in varicells?
That is an interesting question.
I guess if you took the plasma from donor A and mixed it with varicells, would you see the artifact?
I have not done that specifically. I would guess it probably would. I'm not sure. But I will say that I've had some other groups we've been working with who've seen some varicella, also weird varicella artifacts that I suspect are somehow related. They're not exactly the same, but somehow related to this. So I do think that the varicells are potentially susceptible to some of these issues.
And since you did mention Pathsetter, so Pathsetter is a software package that comes with the MDIPA kit. Did you have a chance to run any of these samples through Pathsetter?
Yep, we did. And actually, Pathsetter, it's a little hard for me to QC today. So Pathsetter does identify, it flags kind of when you have too many double positive, because it uses essentially CD3 and CD19 as part of its QC of its multiplet filtering, right? So one of the things that Pathsetter does is it actually runs like an initial cleanup model based on the Gaussian parameters and DNA and like bead removal and things like that. And to QC the resulting output, it actually uses the CD3, CD19 gate to tell you if you have too many essentially to show you the effective removal of multiplets from the sample. So samples that are particularly problematic will be flagged, but it seems to only be if they have extremely high CD19 positivity. So as long as the CD19 positivity on the off-target populations is sort of lower than on the primary population, Pathsetter doesn't seem to really care too much about this issue. But we haven't done this extensively. I've only run it on probably a handful of samples. Yeah.
Okay. So we have about a couple of minutes left. So if you have any last questions, please post them now. Meanwhile, we do have one question waiting. Was there any difference in the staining issue between fresh whole blood samples and stabilized samples?
Yeah, that's another good question. So just as a reminder, the way we're doing our staining here, all the staining is actually happening on the fresh blood. So we're not staining post smart tube fixation. We're staining pre-fixation, and then after the cells have been stained, we're adding the stabilizer to fix them. I think the issue, if we were to stain them post-stabilization, I guess the issue wouldn't exist because generally speaking, we don't do that in the presence of autologous plasma. We usually will stabilize the blood, fax slice or do something to it, wash it, and then stain the fixed blood cells out of plasma. So this is all happening on fresh blood. But maybe another way to phrase your question is, or maybe, hopefully this is what you were getting at, is that if you use fresh blood sort of with the smart tube protocol versus fresh blood with kind of the standard Fluidigm protocol, and we've seen it in both instances there. So we did a head-to-head comparison because that was one of the things that came up was, is this something to do with this modified protocol that we've been using? And we see it using both kind of the conventional workflow as well as the Fluidigm workflow. I think it has I think the extent of the artifact has something to do also with kind of how long the blood incubates and sits around for. I think like relatively small differences in incubation time can have a bearing on this. So yeah, it's definitely a tricky thing that probably warrants more digging into in that regard.
Yeah, and actually that's an issue that I was speaking to a Fluidigm rep and he mentioned that as an option, the difference between the Fluidigm protocol and the smart tube protocol. So I'm encouraged to hear that you managed to reproduce this in the Fluidigm protocol as well.
Yep. Yeah, we don't think it's due to and again, we'd seen some experiments where we thought it was higher in our protocol, and then we repeated it and we saw it was higher in the Fluidigm protocol. So I don't think it's so much the protocol as much as I think because in each of those times, I think one of the samples sat around a bit longer than the other. Because we were kind of doing one workflow while the other one was going on. We didn't track the time perfectly when we did it. But yeah, we've definitely seen it across both protocols.
Got it. So we have one last question. Just putting some context on it, you did mention that you looked at the SomaLogics assay and didn't see any covariates.
Yep.
The question is, did you see any association between clinical phenotypes, such as age, gender, and comorbidities, and the presence of these putative plasma factors? Wondering, for example, if they could be circulating factors such as lupus, anticoagulant, or other immune reactive factors? Yeah,
it's a good question. I have not. I looked kind of cursorily, but I don't think enough to be able to rule out that something like goes on. We are using this in the context, as I mentioned, of this impact study, which is this very large clinical study. It's thousands of samples from across the country. Where the clinical data collection is also hopefully very structured there. So I'm hoping we might be able to ask questions of that sort because we've already seen some indication that this artifact is present in some of those samples as well. So maybe a better handle on that when we have more data to look at.
Yeah, and I think it would be pretty impressive if this work leads to some clinical implications, such as CD45RA reactivity with some sort of medicine or condition that exists out there.
It'd be pretty cool.
Yeah. Okay, that's all the questions. Thank you everyone for attending, and thank you Adeeb for the webinar.
All right, thank you all.
Have a good day, everyone.
Take care, bye-bye.